Are we good to start?
Yes, Avinash, you can start. I think everyone has joined.
Okay.
Yes.
Excuse me, sir. Can I uh, ask something right now?
Yes, tell me now.
So actually, last time we that said command was there, not the last one, and
Yeah.
to that command to change the letters that was the said and in single colon s slash at the red slash that greater than mark was there, so
Yeah.
that was working in terminal, but when we did the permanent change wala hyphen and I, that was not working at terminal. So that is what I get confused, why was that not working?
Yeah, I can have a look at it later if you don't mind.
Sure,
Let's
sure,
start
sure.
with today's lecture and then
Yes, yes,
maybe I
sure. So
can I can have a
okay,
look
fine,
at it.
no issue. Yes, yes,
Okay.
fine. Yeah.
Yeah. So today, I mean I hope we have had a decent understanding of the Linux commands till now. What we will be doing today is we'll be discussing briefly about few of the file formats that are commonly used in the field of genomics. Um, to begin with, let's talk about the most commonly used file format or the one file format which you can't avoid if you are working in the field of genomics. That is known as the FASTQ file format. Okay, so the standardized format for representing sequences in genomics, for representing sequences that come out of a sequencer, the machine, is known as the FASTQ file format. Uh, if you see over here, this is an Illumina sequencer. And if you look over here, these are the various steps that are involved in the whole process of Illumina sequencing. Now, when you look at this picture over here, this represents a flow cell, which is the device on which the entire sequencing takes place inside the sequencer. Okay. So this flow cell has a unique ID. Okay. And within this flow cell, there are 8 lanes. You can see these lanes, right? Okay. Each of these lanes has got tiles. So imagine a graph sheet. Imagine how it looks like. And then superimpose that image on in this flow cell. So each of these lanes have those small blocks which are known as tiles, and the coordinates x and y coordinates of each tile is also noted. It's recorded. So when this entire process of sequencing, which involves all these steps, is taking place inside the machine, when the final output comes out, it has all the information starting from the flow cell ID to the lane of in which it got sequenced to the tile, the exact position from which that particular read is come from, that particular sequence is coming. Okay. Now we might have heard this. Now when we talk about NGS, we don't use the word sequence quite a bit. We use the word read quite a lot, right? Now if you have ever wondered what a read is, Well, read is nothing but a basic unit of data generated from a sequencer, which may or may not represent the entire sequence of your molecule. When I say it may or may not represent the entire sequence of the molecule, that means the biomolecule that you are planning to sequence, be it mRNA, be it the exon, be it an exon, be it the entire genomic DNA, or be it small RNA, the size of these molecules differs. Okay. So if it is a small RNA molecule which ranges from 18 to 50 base pairs, then we can say that one read would represent the entire molecule. But when we talk about something bigger, say for example the 16S rRNA gene in bacteria, that region itself is around 1500 base pairs. So you can't sequence that entire region in one go using Illumina sequencing technology. Or even when we talk about genomic DNA, say bacterial genomic DNA, 3.4 to 5 MB easy, you won't be able to sequence that entire portion in one go. So for that very purpose, I use this term कि it may or may not represent the entire sequence of a biomolecule. Okay. Now there are, so if you look at this figure over here, you have an insert which represents the biomolecule that is about to be sequenced. Now these inserts are surrounded by adapters, or you can say uh, there are adapters that are attached to the either ends of the sequences. Now, these adapter sequences, we'll talk about them in more detail tomorrow. But for today, you just need to know that your insert or the DNA or the RNA molecule that you're planning to sequence is an unknown entity. You don't know the entire sequence of it, like sequence of bases on it. That's the reason why you are sequencing it. So, for it to attach itself on the flow cell like this, you need to fix certain sequences that are known so that they can attach themselves to this flow cell. Now, on this flow cell, you have a bed of oligos. So, ideally, these sequences that help your insert to attach itself on the flow cell should be complementary to the oligos that are present on the flow cell. Okay, so these adapter sequences are nothing but set of sequences that would help your insert, which is the biological molecule of interest, to attach itself on the flow cell so that the process of sequencing can take place. Okay, last thing, if the insert or the biomolecule that you're looking to sequence is large, is big in size, you won't be able to sequence it entirely in one go. Okay, or because in general, beyond a certain point of time, what we have observed with Illumina is that the overall sequencing quality decreases. Okay, beyond a certain point of time. So to be safe rather than to be sorry, what the people at Illumina designed was a bi-directional way of sequencing, wherein you sequence the same insert in two directions. One is 5 prime to 3 prime direction, and the second is 3 prime to 5 prime direction. So the reverse of it. The one which you sequence the read or the sequence that you get as an output from 5 prime to 3 prime sequencing is known as read 1 or the forward read. Okay, and the output or the sequence or the read that you get by sequencing your insert in 3 prime to 5 prime direction is known as read 2 or the reverse read. Okay, so if you have just one read coming out as an output for your insert, you would call it as single end sequencing. But if you have two reads representing a particular fragment, then you would call it as paired end sequencing. Are all these concepts clear to everyone?
Sure.
Yes, sir.
Okay,
Yes, sir.
okay, good. Now, One thing that has to be noted is that we don't just get one read. It's not a single read. Multiple such reads are generated in a sequencing experiment. And all these reads that are generated are together stored in a file which is known as the FASTQ file. Okay. Now, you have 4 lines per read. Okay. So as I said, I will repeat myself again. It's never a single read. There are multiple reads that are generated in a sequencing experiment. All these reads are stored together in a FASTQ file. So if it is a single end sequencing, all the reads would be stored in a single file. If it is paired end, then all the forward reads would be stored in one file and all the reverse reads will be stored in the other file. Together, both the forward and the reverse reads would be used for your analysis. Okay. And when I say read, based on whatever I have shared till now, you might imagine it to be a sequence. Well, it is a sequence, but it also comprises of other set of informations as well. Those informations are distributed in 4 lines. So together, these 4 lines contribute or constitute a single read. Now, before we get into something else, I have a quick question for you. When I say that there are 4 lines per read, if I have 200 lines with me, how many reads am I talking about? Can anyone quickly tell me?
So for pair end or single end sequence?
That wouldn't matter because
50.
pa- paired and I mean the reverse, if it is paired end, both the format won't change, whether it is single end or paired end.
Okay.
Okay,
50 reads then.
50 reads exactly. So I hope everyone got this. How we got 50? Because there are 200 lines, and as I said, 4 lines per read, so we divide 200 by 4, and the option output is 50. Is this clear to everyone?
Yes, sir.
Okay.
Yes, sir.
Before we start practicing it, here is a quick demonstration of how this entire thing would look like. So, if you see, we have two types of FASTQ files here. One is paired end, which has got _1 and _2, indicating the _1 read file is the forward read and _2 read file is the reverse read. And then there is this which says single end dot fastq. Okay. Now remember we had used cat command to display the contents of a text file. Now fastq is a text-based format. Keep that in mind. So you can use cat to display its contents or even head for that matter if you want. So when I did cat single end dot fastq, I got this as an output. So one thing that we need to keep in mind is the first line of a read will always start with at the rate symbol. Okay, it will always start with at the rate symbol. So when I spot at the rate symbol here, I'll start counting. So line 1, line 2, line 3, and then line 4. Then again I get an at the rate symbol in the next line. And the format of this line is pretty much similar to this line, right? The only difference is there's 5 here, there's 2 here, and then there's a difference in these numbers. So this means this marks the beginning of the second read. So 1, 2, 3, 4. This is where my second read ends. Now, does this give you a better picture of how the reads look like?
Excuse me, sir. I lost my network. Can you repeat this slide once more, sir?
Yeah. So what I was trying to say here is that we have three FASTQ files here. Single end fast queue, as the name itself suggests, is has the output or reads coming from a single end sequencing experiment. Paired underscore one and underscore two dot fast queue indicates that the file which is labeled as underscore one will have all the forward reads, and the file that is labeled as underscore two will have all the reverse reads. Now, how do the reads look like? You have again, FASTQ is a text-based format. So if you look here, I have used cat command to display the contents of single end.fastq file. So as I had said previously as well, cat command is used to display the contents of text-based files. So you can use cat to see how your FASTQ files look like. Even head and tail would work here. So if you look, the first line starts with at the rate. So one thing that you need to remember is in FASTQ format for every read, okay, in every read, the first line should always start with at the rate. So this is my line one, this is my line two, this is my line three, and then this is my line four. When I come to line five, I observe that it's starting with at the rate plus this format is pretty similar to the format of the first line that I had seen, right? So this would indicate that I am moving on to the next read. So this would be read 2 line 1, read 2 line 2, read 2 line 3, and read 4 line 4. So together we are looking at two reads from single and fast queue file. Is that clear to everyone?
Yes, sir.
Um, the
Yes.
participant
Yes,
who had
sir.
lost
Yes, sir.
Yes.
his network, is that clear?
Yes, sir.
Yes.
Thank you, sir.
Okay. Now, it might look as a strange combination of alphabets and numbers to you, but each line has a meaning. Okay. So when I start with the first line, this line is known as the sequence identifier. Now remember, if you try to remember, we had name slips on our notebooks and registers when we used to go to the school. Name slip had everything: our name, our class, our roll number, and then the subject to which that notebook belonged, right? Consider this sequence identifier as that name slip. So the first line is known as the sequence identifier, and just like the name slip, its function is to trace down the entire address of the read. Now, if you remember, I had told you that the sequencing inside the sequencer takes place on a flow cell. Flow cell has its own unique ID. It has lanes, it has tiles, and the coordinates of each tile is recorded. Keep that in mind when we discuss about sequence identifier further. Okay. For now, just to be familiar, what I'm telling you here is that the first line of any FASTQ file, any read in a FASTQ file, would be a sequence identifier. The second line looks more familiar. This is the sequence. So this is the sequence of the template that we had sequenced inside. The next symbol that you see, the plus symbol here, is known as the quality header. Now, previously, the same information after at the rate, I mean, they didn't use at the rate. Whatever you see here after at the rate was repeated after the plus symbol. But over a period of time, the people realized that this is redundant. This is just adding extra information to your file, size wise, right? It's not adding anything new, but this is just adding extra size to your file. So they decided to stick with the entire sequence identifier information in just the first line of the read and decided to omit it from the third line. Now the purpose of this plus symbol is not much. It's just a placeholder, and it just marks the point of separation between the sequence and the quality score. So I think I have already given it to you guys. The fourth line here represents the quality score of each base that has been identified at the time of sequencing. So for base A, my quality score is O. For base G, my quality score is T. For base A, my quality score is S. So on and so forth. Is this clear to everyone?
Yes.
Yes, sir.
Okay.
Sir, uh,
Yes.
uh, sir, these alphabets have a specific value. Yeah, in
Yeah,
which
yeah, we are
sentence?
coming to that. We are coming to that.
Okay, okay.
I'll make you guys do a small exercise as well. So we are coming to that. So let's start with, so the first, I would say, cryptic thing here was the sequence identifier, because if you just read it, it won't make any sense at that point of time. So there are a lot of things that you can understand from this line. At the rate starts a new sequence, right? As we had discussed previously. This NS500123 is the instrument ID. Oh, okay, the ID of your instrument. Run ID. Now there are multiple runs associated with the sequencer. So, which means the number of times you are running your entire sequencing experiment inside the machine. So the run ID for this particular experiment was 73. Then remember I had shown you that you have a flow cell ID. So this, the number that you see here, the set of characters that you see here represents the unique flow cell ID. Each flow cell has its own unique ID. Now I had told you there are lanes on a flow cell. There are 8 lanes on a flow cell. The number 5 here represents the lane number. Then I had again told you to imagine a graph sheet and superimpose it on your flow cell, like you have blocks on your graph sheets. Similarly, those blocks will be present on your flow cell as well, and they are known as tiles. So this 1208 represents the tile number. Okay, then x and y coordinates of the exact position from where this particular read is coming. Okay, and read number 1. So if it was a single end experiment, you would have 1 here. If it was forward read of a paired end sequencing experiment, you would still see 1 here. If it was a reverse read of a paired end sequencing experiment, this 1 would change to 2, indicating that it is a reverse read. Okay. Now there is a filtered flag which indicates whether the read has been subjected to any quality filter or not. Okay. Generally, they don't do that. You don't do it by default. Filters are like quality control is done at R and at an informatics level. Okay. So N here means the read hasn't passed quality filters, but that does not actually mean that the read is bad. Till the time we don't subject it to any quality control protocol, we won't say that it's a good or bad read. Then control number is 0, which indicates that there is no control used. If you have used a control, then it would have been 1. Then index sequence. Now, previously, what used to happen was that you need to have enough samples to fill the entire flow cell. Otherwise, it would be very expensive. Like if we talk about RNA-seq experiments, back in the day when sequences were new, it would cost anywhere between 1.5 to 2 lakh rupees to sequence one RNA-seq sample. Now you are talking about rates in thousands, right? 15, 16, 17, I don't know what the latest rates are. But back in the day, I'm talking about 2010, around 2008, 2010 k around, it was really expensive. Why? Because it's just like public transport. If you have a single person sitting inside a bus or a minibus, or if you have 50 people occupying the bus, the cost of operations would remain the same. Here also, you needed to have, if you had single sample, you would end up paying the same as you would pay when you had multiple samples. So what they decided was that let's use certain barcode sequences and load multiple samples from different experiments inside the same machine. And using this barcode index, you could then later segregate your samples from the lot. So that is what this index sequence here indicates, designates. This is your barcode using which you would segregate reads from your experiment from the combined lot. So this ensures that data doesn't get mixed. Okay, is that clear to everyone? Is this clear to everyone?
Yes, sir.
Okay,
Yes, sir.
okay.
Yes, sir.
I'm just taking it as like, I mean, this whatever yes or no I'm hearing, I'm taking it as a representation for the entire batch. In case if there is an issue, you can type it in the chat section as well. I guess the moderators would be more than happy to convey it to me. Okay. Now, I think there was a question asked, do these characters stand for a particular score? How do you interpret what the quality scores would look like? So there's something called as ASCII, which means American Standard Code for Information Interchange. This table that you see here, the ASCII table, and each character that you see on the decimal side here, sorry, on the character side here, has a corresponding decimal value which we use to calculate the quality scores. Okay, now, for example, my first base is A and its corresponding quality score is O. So I will look for the value of O in this presentation. So the value of O in decimal is 79. Okay, perfect. Now we measure temperature for like it's an example. Don't get confused as to where this thing came in between of reads and quality scores. But when we measure temperature, we have temperatures in degrees Celsius, we have temperatures in degree Fahrenheit, and we have temperatures in degree Kelvin. Now, if we have to convert temperatures from Celsius to Kelvin, what do? We just add 273, right? So if my temperature is 30 degrees Celsius, I would add 273 to it and it becomes 303. And when I have to do the opposite, as in degree Kelvin to degree Celsius, I subtract 273 from it. So 300 minus 273 again becomes 27. So whatever the temperature is, that would be in Celsius. So 273 is the base. It's the base value. Here, that base value is 33. Because if you look at the table here, all the characters before decimal 33 are either space, escape, substitute, cancel, shift out, shift in, enter, tab. What does that mean? That even if you press these characters, for example, if you press shift, there is something changing in the backend, right? The computer is working, doing something in binary and making the system understand that the next letter that we type would be in capital. But when you press shift, does anything change on your screen? It doesn't, right? Similarly, when you talk about tab, your cursor shifts, but do you see anything on the screen? You don't. Similarly, enter. When we press enter, our line changes, but do we see any character on the screen? We don't. So everything before decimal 33, that is exclamation mark. All the characters before that indicate blank spaces. So we don't want blank spaces as our scores because then it would mean 32 different combinations, right? So the first character that can be comprehended visually on the screen when typed on the keyboard is exclamation mark. So we use its decimal value, that is 33, as the base. Okay. So whenever we are in the process of calculating quality scores, we would subtract 33 from the value of the character, and the number that we get would be the quality score. Is that clear to everyone?
Yes, sir.
What about the rest? Is it clear to everyone?
Yes, sir.
Okay. So quickly, as we said, A, the base, the quality score is O, capital O. Remember, small and capital mean different here. So I look into the table, the value of O is 79. I subtract 33 from it, and my quality score is 46. Now, this is the real quality score. What does it mean? If my quality score is 10, the probability of incorrect base call is 1 in 10, and my base call accuracy is 90%. If my quality score is 20, my probability of incorrect base call is 1 in 100, and my base call accuracy is 99%. If my quality score is 30, my probability of incorrect base call is 1 in 1000, and the base call accuracy is 99.9%. So on and so forth. So higher the quality score, lesser is the probability of incorrect base call, and higher is the base call accuracy. Is that clear to everyone?
Yes, sir.
So which quality score, like from which quality score we should consider it as correct one?
That's a good question. So generally, it all varies. Initially, as I said, in the initial days of Illumina sequencing, your quality score of 20 was considered to be brilliant. Okay.
Okay.
Over a period of time, as the sequencing protocols improved, we have now increased the standard to Q30, which means a quality score of 30 or more is considered to be the best.
Okay.
Aajkal, nowadays, we would prefer Q30 or more. Is that clear?
Thank you, sir. Yep.
Okay. So, is FastQ format clear to everyone?
Yes, sir.
A quick yes or no before we
Yes,
move
sir.
on.
Yes, sir.
Yes, sir.
Perfect. Good. So, this is the first sequencing format that we wanted to cover today. We'll move on to the next one. I'm stopping my screen share for a bit. Is my screen visible?
Yes.
Yes, sir.
Perfect.
Yes, sir.
But I have lost my cursor. Oh, it's here. How many of you have heard about the FASTA format before?
We know it, sir.
Yes, we heard.
Yeah.
So, do you know about it?
So in PubMed, whatever the sequence we get, So certain accession code, and along with that, you'll get the sequence. So that we call in faster format ATGC sequence.
Correct. So regardless whether you know it prior to this workshop or not, let's discuss about it. So it was proposed in 1985 by the two gentlemen here, David J. Lipman and William R. Pearson. Now, it's a text-based format for representing either nucleotide sequences or peptide sequences. So it's not just nucleotides. You can store amino acid sequences as well in FASTA format. Now, if you remember our FASTQ, in our FASTQ files, the read will always start with @ symbol. In FASTA format, this is slightly different. Your read or your sequence will always start with a greater than symbol. Okay. Now, FASTQ doesn't have a limit on the number of characters that can be included in a file, in a line. Okay. Here, there is a limit. You cannot have more than 80 characters in a line, including spaces. So, what you see here, as someone was rightly pointing out, you have accession number and then ATGC. Here it's not ATGC, here it's amino acid sequences. But in this line as well, you have this accession number and everything. It can't go beyond 80 characters. Ideally, it should not go beyond 80 characters. That's recommended. If you look at it, I'm pretty sure it's more than 80, but like you have good lab practices, good informatics practices के according, this character length should be less than 80 characters. Okay. Also, you should not have any numerical digits, any numbers as part of your sequence. You can have your numbers as part of your first line. Okay. You can have numbers as part of your first line, which is the description line. Okay. But numbers should not be a part of your sequence line. Anything that is unknown can either be represented by N in case of nucleotides and X in case of amino acids. Okay. And why are we talking about it? Because in RNA-seq experiments, we need to align, we need to compare our reads against a reference genome. And that reference genome is always in FASTA format. So whenever you see a file of this format, you should know that this is in FASTA. Is that clear to everyone?
Yes, sir.
Again, unlike the FASTQ format, there is no such rule of 4 lines per sequence or per read. You can have as many lines in a for a read or for a sequence as possible. The only criteria is that all the nucleotides or the peptides or the amino acids should be covered for that particular sequence. Lines आपकी जितनी मर्जी हो, that doesn't matter. The upper limit is just on the number of characters. So 80 characters per line. Is that okay?
Yes, sir.
Okay. So this is a short exercise which you can work on later as well. So as we know, grep gives us the count of number of characters in a file. And if we add -c to grep, it gives us the count like in numbers. Grep without -c will just fetch those sequences with those lines which have that particular sequence or that particular character. And if we add -c, it would give me the count. So when I add -c after grep and look for number of greater thans in my file, that would give me the number of sequences. Okay. So you can also try it when we do a quick practical of it. If you count the number of occurrences of greater than symbol in your file, that would be the number of sequences in the fast file. Okay. The next format that we are going to talk about is the GTF/GFF format. Um, it was designed in the early 90s, and GFF was designed in early 90s and GTF in 2001, and it was proposed by the European Bioinformatics Institute and Wellcome Sanger Institute. Both of them were like part of the Human Genome Project as well. So if you look here, you just see a sequence of ATGCs. You don't know from which position to which position there is an exon. First of all, you don't know whether it's a gene or not. You don't know whether it has any exonic regions or not. You don't know whether within exons there are any coding sequences, CDSs or not. Where is the UTR, 5 prime, 3 prime? All these things we don't know, right? For us at this point of time, it's just a plain sequence, right? So for that purpose of refinement, we needed a file that would tell us key from which position to which position we have a particular feature on our genome. Okay, when I talk about feature, it can be an exon, it can be a CDS region, it can be a UTR. So whether if we have any of those features on my sequence, that information is conveyed by the GTF file. So again, all the file formats that we have talked about till now, they are all text based. Okay, all of them are text based. So this is how a GTF file, this is a snippet of how a GTF file would look like. There are 9 lines or columns, okay, in a GTF file. Each of these fields or each of these columns are tab separated. So between two entries, the separator is a tab. Every information related to your genome or a gene or a sequence can be extracted from its corresponding GTF file. Now let's see what each of these fields or columns stand for. So the seq column that you see here represents the chromosome, or yeah, it represents the chromosome. So for example, this is say 381, or if it was a human genome, it would be say chromosome 9, chromosome 10. So you would have CHR9, CHR10 written here. Like for example, here you have 1, so it's chromosome 1. Okay, this represents the chromosome. Okay, second column is the source. So when I say the source, I mean the program that was used to identify the features that are present on this sequence here. So the program that was used for
Yes,
that
sir.
purpose.
I'm sorry to interrupt, but that 381, how come it will be chromosome? I did not get that.
Okay, so is the screen visible here?
Yes, sir.
So this is another example of a GTF file. Do you see 1 here?
Yes, sir.
This one is chromosome 1. Okay, this is a snapshot of the GTF file for rat genome.
Okay.
Okay, now it depends on how what annotation format has been chosen for that particular genome. So Arabidopsis genome will have a different format. Rat genome will have a different format. Human genome has a different format, so on and so forth. So, whatever format you have chosen to represent your chromosomes in, the same format would be present in the GTF file. Does that make sense?
Okay. Yes, fine. Yeah.
Also, this 381 can not only be the chromosome, it can also be the scaffold, because sometimes you don't have chromosome level information. So what you do, you just pick that scaffold from the genome and annotate it. Okay. So whatever feature has been annotated, be it a chromosome or a scaffold, that number would be in the first column. Is that clear?
Yes, sir.
Okay. Moving on. Source, as I had said, is the program or tool that was used to predict this GTF file from the genome. Okay. Third column talks about the feature, whether it's an exon, gene within that gene, whether it's a CDS, whether it is a start codon, stop codon, so on and so forth. Start and end would represent the starting position and ending position of this particular feature. Okay. Score in terms of base pairs. So, for example, if I have a CDS from position 10 to 20, so it will count. Okay, if this is 10, so this will be CDS1, and then it will continue till 20, and then this would mark the end of CDS1. Then subsequently there could be another feature or an overlapping feature, so on and so forth. Okay. Next, we have score. If present, it is just a prediction score of how accurate this feature, how accurately this particular feature has been predicted. And if this is not present or not available, you just put a dot here. Dot means that this feature is not available. Then you have strand. Strand, whether it is on the sense or antisense strand. Then you have frame, which means whether the start codon is also the first, like whether it's the first codon of the feature. Yeah, whether the first. Okay, so if it is zero, that means, for example, if a feature starts from this C, okay, if this C, the one that I have highlighted here, is the start codon, is the first base of the start codon, then your frame would be zero. If your feature starts from here, but the second base here, like the third base here, is the first base of the start codon, then it would be 2. Or if the first feature here is the base of the start codon, then it would be 1. Okay. And the final feature is the annotation attribute, wherein extra information about this particular feature is present. For example, it would give you the gene ID, it would give you the transcript ID, it would give you whether it is a protein coding gene or not. So all that information would be present in the attribute column. Okay. So these are our main columns, the 9 main columns. This is our attribute column. So all the information that can be present in this particular column is, you would have your gene ID, you would have your transcript ID, you would have the gene name, gene type, whether like transcript type, exon number, exon ID, description of this gene, like what exactly does it do, the function that this particular gene performs. So yeah, so this is our GTF file. Any doubt till now?
No, sir. Fine.
Everyone, is it okay?
Yes, sir.
Yes, sir.
Okay. Now, there are few exercises in as well which we can practice later. So we are done with GTF, GF, we are done with FASTA, we are done with FastQ. Now the final for file format relevant to this particular analysis is the BAM format. I'll need a minute. Is my screen visible?
No, sir.
No, sir.
Okay. Uh, is it visible now?
Yes.
Yes, sir.
Yes, sir.
So, yes, sir.
Okay. Now we have another format which is quite important, that is the SAM and BAM format. Now, SAM stands for sequence alignment mapping and BAM stands for binary alignment mapping. These formats were developed by Heng Lee and Bob Handshaker in 2009. So it's a fairly recent format as compared to the other three rather. So one thing that I would like to clarify. Oh, do you have a question? I see someone has raised a hand. Rituparna, do you have a question or it's
No, sir. I just raised my hand
oh,
earlier.
okay.
I think. Yeah.
Okay. So yeah, so both these formats contain exactly the same information. SAM is text format and BAM is compressed SAM format. So whatever information is present in SAM, its compressed version is present in the BAM file. Okay. Now there are two sections of a SAM or BAM file. One is the header section and second is the alignment section or the alignment information section. Now, the header section, it contains regular information like SAM version. It will tell you whether the file is sorted or not. It will tell you information about the reference sequence, so on and so forth. So it's basically just a build up to the file. The main information is present in the alignment section. Now the structure of the SAM or BAM file is similar to the GTF file, as in you have different columns and each column kind of follows a similar format. So if you look at the first column here, it says read ID XYZ/1. So this represents, any idea what it would represent? Any guesses?
File location or something.
Not exactly. Any more guesses?
They're similar to the first line of fast queue.
Perfect.
Right.
So this would tell me which read is being aligned against the reference here. So this would be the first line of my fast queue file. That would tell me ki, okay, we are talking about this particular read. Is that clear? Now,
But the at the rate symbol should be there or no? I mean, I'm not
that's
understanding.
here it's not needed because it's not a fast queue file, right?
Okay, okay, got it. Yeah,
Yeah, so that is compulsory if you are working on a fast queue file. This is
fine.
just information from the fast queue file, so you don't need at the rate here.
Okay, thank you, sir. Yeah.
Nice. That was an interesting point of view. So good. Shows that we are attentive. Um, okay, we'll skip the second column for a bit. Come to the third column. What do you think it would be? Just guess. कोई marks नहीं मिल रहे हैं इसके.
Number of bases.
Third column, third column.
Doing something, I don't know.
Okay.
Adapter? No.
नहीं. No, 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 not adapter. The okay. So the third column here. Acha, what are we aligning our read against? We are aligning it against a reference, correct? So reference में कहाँ align हो रहा है? Where it is aligning in the reference, that information would be present here. Okay. Now I think this would give you a better understanding. Why is my Remember we had this confusion here in the GTF file key, what is this exactly? So as I said, this is the scaffold number or the chromosome number. So if it would have aligned to chromosome 381 or in this case chromosome 1, so here you would have seen 381 or 1 depending on which chromosome it would have aligned to. Does it make sense now?
Yes, sir.
So this is for the read and this is for the reference. Okay. Moving ahead. अभी मैं आऊँगा second column पे. I'll come back to the second column. But I'm just trying to correlate it with the GTF file. So based on whatever you have learned from the GTF file, what do you think the fourth column would be? Acha, by the way, do we know what alignment is? I'm sorry, I should have asked it beforehand. Do you know what alignment is?
Yes, sir.
Yes, sir.
Okay, can any one of you let me know what exactly is alignment?
So it's comparison between two sequences.
Correct. So comparison आप कर रहे हो when you are comparing. There is one reference and then there is one query. Correct? So references are reference genome which has all these chromosomes or contigs, whatever you would call, and then queries are read in FASTQ file. So this number in the fourth column would indicate the starting position of the alignment, from where alignment would start. So we know it is starting in chromosome in contig 43. We know it's this read, but contig 43 may from which base position the alignment is starting, that is 6,13,960. So उस particular base position से my alignment of this read on this particular contig is starting. Is that clear? Is that clear
Yes,
to
sir.
everyone? Okay.
Yes.
Now let's come to the second column. Second column is known as the bitwise flag. Okay. Now this column tells you the nature of alignment. Okay. Through this column, you have to like break it down. 256 in itself doesn't mean anything, but when you break it down into fragments, that is when you will realize whether your alignment is proper or not. Okay. So that information is covered in the second column. One thing that you need to notice is when you break it down, if you get four, okay. So let's quickly do one thing. I'm dropping a link in the chat. All of you open that link quickly. Did you guys get the link? Did you get the link? It's in the chat.
Yes.
Yes,
Yes.
sir.
Yes, sir. Yes, sir.
Yes, sir.
Can you see my screen? Hmm. यहाँ पे if I put 256 and click on explain, so it says not primary alignment. Okay. So this is not the primary alignment, this is a secondary alignment. Then if I put 99 and click on explain, that means tell me read is paired, read mapped in proper pair. That means first the forward read aligned and then the reverse read aligned. If the opposite happens, then you will see read not in proper pair. Yeah, that like if this particular column is not checked, that would mean that your read is not aligned in a proper format. So your reverse read would have aligned before the forward read, which is quite a common when translocation events happen. In that case, your reverse read might align before the forward read. Okay. So yeah, read mapped in proper pair, made reverse strand. So your reverse read is aligning in the reverse strand. So yeah, 99 as a flag would indicate that this particular read is properly aligned. Okay. Then there are multiple flags for proper alignment and multiple flags for improper alignment. One thing that you need to show that you need to know is if you have four anywhere in the breakdown of your SAM flag, because if you would have noticed 99 जो है 99 is not just 99, it is a combination of these many flags in the summary section, right? Can you see that? So if you just have four in your breakdown of the SAM flag, okay, that would mean that your read is unmapped. So when you so if you break down your SAM flag section, if there is four anywhere in its breakdown, that would mean that this particular read is unmapped. So whenever you have a flag, you like in the second column, you can come to this website and see what kind of alignment does it indicate. So we did 256. Let's do 272. Let's see what 272 means. Again, reverse strand, not primary alignment. Do we have any primary alignment examples here? So 99, as I said, proper alignment है. Let's do 88. So 88 says made unmapped. That means this read is unmapped one. That is what I was trying to say. If you have 4 or 8 in your flag section, that would tell you that your read is not properly aligned. And you don't, there's no need for you to remember it, because if you want, you can always come to this resource and check what your SAM flags denote. Is that clear to everyone?
Yes, sir.
Yes, I think so clear, but this link is not opening and I'm not understanding क्या issue है. Explain SAM flag 404 error दिखा रहा है same link.
Can you Google
अच्छा, yeah, SAM
this,
flag.
exactly this line? Explain SAM flags. Just Google it, and the first link that you will get should be from Broad Institute.
Got it, sir.
इसमें you can put your SAM flags in and check.
Got it, sir. Yeah.
Okay.
Yes, sir. Thank you.
Mhm. So we are done with 4 lines. Next is mapping quality. So like we had quality scores, similarly we have mapping quality scores in BAM files as well. So the higher the value, the better the quality is. Okay. And yeah, the one thing, and this mapping quality score is, there's no standard to it, because different alignment tools have different ways of assigning quality scores, mapping quality scores. So we don't have a fixed standard here. But if I remember correctly, the one standard is that if you have a value of 255, that means you don't have an assigned quality score to your alignment. 255, that means that there is no assigned quality score to your file. Other than that, the standard rule of thumb is higher the quality score, better is the quality of alignment. Now, what that highest value would be differs from alignment tool to alignment. Like we'll be using HiSat, it has a different way of grading alignments. There is another popular tool called Bowtie, it has a different way of grading alignments. We have another tool called BWA that has different way of grading alignments. Okay, is that clear?
Yes, sir.
Okay. Next. Next we have the cigar string. Now what does cigars stand for? Well, the full form is a bit too much, but it's concise, idiosyncratic, gapped alignment report. That's the full form of cigar. Now, it is a sequence of numbers and letters that indicate the continuity or discontinuities that you will find in alignment. So each character used in a cigar string means something. So M is for match, X I guess is for mismatch, I is for insertion, D is for deletion. So here what we have observed is that there is a number 65 before M. That means we have 65 continuously matching bases. If it would have been something like 60M, 2X, 1I, that would mean 60 matching bases, 2 mismatches, and 1 insertion. So it basically represents the pattern of alignment. While your flags represent the nature of alignment, whether it's proper or not, whether the reads are mapped or not properly, all these things, this represents the process of alignment. How, whether all the bases matched or not, whether there were any insertions or deletions or not, so on and so forth. Is this clear?
So will you please explain one more time?
Okay. So cigar mein there are certain set of characters that one need to remember. M is match, D is deletion, I is insertion, and nowadays they have included X as well, which means mismatch. So for example, अभी ये मैं आपसे पूछ रहा हूँ, suppose my sequence is 30M, 5D, and 30M. What do you think that would mean?
So 30 matches है, 5 delete हुआ है sequence और then again 30 match. Is that right?
Perfect. So here you have 65M, that means continuously all 65 bases found a matching base in the reference. This is with respect to the reference. Ki in my query, I could find when the alignment started at this position, I found 65 matching bases continuously in the reference.
Okay, so
If
fine.
the
Got it.
pattern would have been different, say for example 30M, 5D, 30M hota, so then it would have been in this context. For this particular read, when I started aligning from this position, I found 30 matching bases, then 5 mismatches or deletions, sorry, not mismatches, 5 deletions, and then again 30 matches. Is that clear?
Yes, sir. Thank you.
Okay. Now, now this particular column talks about the position of the mate. Mate say, what do we mean by the mate? By mate we mean reverse read. Now this is your forward read. Its reverse read would also align somewhere, right? So if that information is available and say for example this read is aligning to the same contig, its reverse read is aligning to the same contig, then you would see an equal to here, which means that both the forward and the reverse reads are aligning to the same reference. If that information is not available, then you just put a star in here. If it is aligning to a different contig, you would put the contig number here, and then that would mean that both the reads, forward and reverse, are aligning to separate contigs. Okay, so if it is equal to, that means they are aligning to the same contig. If there is a different contig name, then that would mean that it's aligning to a different contig. And if it's dot or star, that means there's ambiguity about whether the reverse read is aligning or not. Okay, then you have this column here indicates the position of the alignment. So when you have established that the reverse read is aligning somewhere, then you would also want to know the position of the alignment. So you, like you have here, so base number 6,13,960 pay, this read, the forward read was aligning, started alignment against the reference contig 43. Here, if you have that information, then you would have a similar number in the next column as well, which will tell you from the position from where the reverse read starts its alignment. Because this information is not available, we have 0 here. And using this number, which marks the beginning of alignment of the forward read, and this number, which marks the beginning of alignment of the reverse read, we can calculate template length, the total length of the template. But because that information is missing in this case, we have 0 everywhere in these two columns. Is this clear to everyone?
Thank you, sir. ठीक है. Yes.
Others, is it clear to everyone?
Sir, I have a doubt.
Yes, please.
So actually there is an inner distance, right, in the
Yeah, yeah, yeah.
fragment length. So how we get the that distance, like apart from the reads?
So, are you talking about with respect to the alignment, or are you talking about
No, with respect
in
to the
general?
alignment. No, with respect to the alignment.
Okay, so we so the inner mate pair distance that was asked here. I don't know whether I have it in this slide or was it in a separate slide.
Actually, I think it was in the first.
It was in the first FASTQ slide, yeah. So
Yeah.
basically, it is the distance between the forward and the reverse read. Okay.
Yeah.
So based on where your reads align, so your inner mate pair distance basically regulates whether your alignment is proper or not. For example,
Hello.
yes.
Yeah. Okay.
So this is my inner mate pair distance.
Yeah.
And you can call it as the distance between 3' end of both the forward as well as the reverse read. Now, if so, in ideal scenario, when you are aligning, if this is your forward read and it aligns from base position 10 to 100, okay, and the inner mate pair distance is 50 base pairs, the reverse read should ideally align 50 base pair apart from the forward read, okay,
Yeah.
and the total should be equal to your template length. So you can calculate ki okay, theek hai, I know my read length is 150 base pairs paired end. For my whole template length is coming out to be 350. Okay. So I,
Yes.
matlab yahan se, if I know my template length is around 350, I know my read length, so I can calculate my inner mate pair distance. Does that make sense?
Okay. Okay. Yeah. Yeah.
Is it clear?
Yeah. Yeah. It's clear.
So, based upon the example which you have given, if we have like 100 read 1 and 100 read 2, then if you have 350 base space, that means the inner mid pair distance should be around 150.
Yes, yes.
Okay, got it.
But the problem here is that if your inner mate pair, so this template length, okay. So if there is an insertion or a deletion in between, then there is a high probability कि your reverse read would not align within this distance.
Okay.
So it might align a bit farther than your inner mate pair distance. So that time your alignment will not be, so this, that's why there are two categories: concordant alignment and discordant alignment. Concordant is when your alignment is proper, as in first your forward read aligns, then within that intermediate distance ke gap mein your reverse read also aligns. So that is your concordant alignment. Concordant means something that obeys rules.
Mhm.
But if there is an insertion or deletion event happening or even translocation, so there could be a possibility that your forward read is aligning here, say 0 to 10, but instead of, suppose this region gets deleted, so instead of aligning within 50 base pairs, your reverse read aligns 350 base pairs apart from the forward read. So that time your template length will reflect that.
Okay.
Based on that, you can figure out whether it's a concordant or a discordant alignment. Is that clear?
Okay, sir. Clear. Thank you.
जिनका original question था, is that clear to you?
Yeah, yes, sir. It's clear.
Okay.
Thank you.
Yeah. So, so this was my template length. Any idea what the next two columns have? This column and this column? So this is
So
my
the
sequence.
sequence and the score, quality score.
Exactly.
Yeah.
So this is my sequence of the read, and this is its quality score. So that's the information. And the final line, which is this, just like the attribute column, these lines represent some additional information about my alignment. Okay. Now, for example, as I said, we have concordant, discordant alignment. Mm, it's UU. I'm not pretty aware of what UU here means, but जैसे ये particular tag है. If you look at this tag, YT colon Z colon, if it would be CP, that would mean concordant alignment. If it would be DP, that would mean discordant alignment. Then there is mismatches as well. Yeah, NMI 0. If you see here, that means there's no mismatch. If there would have been one, that would have meant one mismatch. So likewise, each of these tags has some meaning or the other pertaining to the alignment. And most of the times, these tags are used by quality assessment tools to give you additional information about how your alignment looks like. Okay, so this is basically your SAM or BAM format and how it looks like. Now, I hope all the file formats discussed till now are clear to everyone.
Yes, sir.
Can I get a quick yes or no from everyone in the chat? If
Yes,
you're
sir.
not comfortable speaking, yes or no in the
Yes,
chat would
sir.
help.
Yes.
Okay.
Yes.
Now, oh, okay. Now we'll do a quick practical. Okay. So I hope all of you have downloaded all the files that Dr. Jibin had shared with you, like that
Yes,
link,
sir.
the Google Drive link. Do you have it?
Yes, sir.
Can anyone of you tell me where did you download it? I mean, is it in your
Sir, it's,
desktop?
yes, sir, it's in desktop.
Yes, sir.
Okay. So open your terminals. I won't commit that mistake this time. I'll transfer it to the desktop. Just give me a second. Is my screen visible? It might not be. Is my screen visible to everyone?
No,
No,
sir.
sir.
Is it now?
Yes, sir.
Second. Okay. So, can you all come back? Okay. Uh, so what we need to do right now is to edit this. Okay. To start with, I'm sorry, I just skipped the gun. All of you are in desktop? Are all of you on your desktops? Like on the terminal?
Yes, sir.
Hello. Okay. Can you type PWD? Done?
Hello, sir. Yeah.
Yeah, can you type PWD
Uh, no, sir.
from
We need to
desktop?
first go to the desktop first.
Yeah. Is it
Yeah.
pasted on your desktop? Is the folder
No,
in
no.
your desktop the
Yeah,
one which you downloaded?
yeah. I have the folder. I'm asking in the
Okay, so
terminal.
go to
First,
desktop.
Okay, okay.
Sorry?
Yeah, yeah. I got it, sir. Thank you.
So,
Okay,
Mr.
if it
Abhinav,
would
sorry, if
it
I'm
is
if I
CD.
downloaded the file
Sorry?
within, if I have downloaded the file within the downloads folder, so uh I'm gonna go within the file, not on the desktop.
You can, instead of desktop, you can go to downloads, or maybe
Okay.
you can paste the file on your desktop and then do the same. It's whatever
Uh,
is convenient to you.
yeah. So I'm telling
Okay.
you. So I'm just telling you that I'm within the downloads because I have downloaded the file within the downloads.
Okay, works. But just keep in mind that whatever command that we type here, then you need to alter it as per your
Exactly.
folder,
Yeah. Yeah.
folder's position, right?
Right. To the path.
So just keep that in mind. That's all.
Okay.
So it's MNTC users, Avinash, OneDrive, desktop. So instead of Avinash, it would be your username. Are all of you done? Are all of you on desktop? Guys, yes or no, please?
There's coming no such file. I put
Yes,
it on
sir.
desktop, but still it's not showing no such file on the desktop. I might be being wrong
Can you
command. Yeah, just a moment.
can you share your screen, please?
So this mnt/c/users/prachikapse/desktop I'm putting.
This won't work. This is MacBook. I'm sorry. Type CD.
CD.
Where did you copy it? Can you show it to me, like on your
Yeah, on
like
my
on your screen?
desk.
Can you show me where did you copy your file?
Here only. These RNA-seq reference
It's right
and
here. Ye jo whatever I'm
yes,
seeing in front. Okay.
yes.
Can you
Okay.
open the terminal?
Yeah.
Can you delete CD?
Sorry, delete CD. Hello,
CD हटा दे. Yeah, CD delete, please.
CD delete.
Type
Okay.
ls.
LS.
Yes.
Okay.
Type ls. Yeah.
Yeah.
Type
So
CD desktop. There's a
okay.
desktop right here, no?
Yes.
D
Yeah.
is capital, man. D is capital.
But
Check how it's written.
should I do LS here?
Okay. Type ls. Type ls.
Yeah. Yes, sir. I can see the RNA reference based file here.
Perfect. Good.
Okay. Thanks.
So, so all of you, if you are on desktop, type pwd and copy this path, please. Copy it. Just select the entire thing and press right click on it so that you can copy it. Done. Have you copied it, guys?
Yes, sir.
Okay, just right click here to see whether it has been copied or not. Can you paste it on your terminal?
Yes, sir.
Okay, cool. Now type ls. If you can see RNA seq reference based, type cd RNA and press tab so that that entire thing comes up. Press
So we
enter.
can see your screen, sir.
So
Then type ls.
Sir, screen to dikha.
Can you see my screen now?
नहीं समझ में आ रहा है.
Can you see my screen? Is it is it visible now? The screen?
No, sir.
Yeah?
No, sir, it is not visible. Yeah, it is visible now.
Oh,
no,
sorry. Now I think you can see my screen now.
Yes, sir. It's visible.
Okay, so what I'll just repeat whatever I did till now. I typed pwd from desktop. I got the path. I selected it and then did a right click so that it gets copied. Then I typed cd rna and then tab key. Type rn, press tab, so it will automatically fill up RNA-seq reference paste. Press enter. You should see RNA-seq reference prompt. Are we all done till this step?
No, sir, you are
Sir,
going
I'm
very
having an
fast. I'm
issue.
not able to understand.
Okay. Are all of you on desktop?
So we
No,
I'm
sir.
just the first step desktop. After that, you have gone very fast.
I'll repeat. I'm sorry. I'll repeat that. Now, can any one of you share your screen, please?
Mhm.
Wait. Wait. So, I suppose all of you are on desktop, right? Everyone. I need, तभी मैं बोल रहा हूँ. That's why I'm saying just
No,
let me
sir.
know if all of you are
Yes,
listening
sir.
or
We are
at
in
the
the
same page or not. That would help me streamline.
Sir, I'm
I
yes,
heard a
sir.
no in
I'm
between.
facing an issue.
Who said no?
Shikha.
What is the issue?
Sir, can I share my
Yes,
screen?
what's the issue? Of course.
Sir, I'm not on desktop, I guess.
Where have you copied this thing? Where did you copy the folder? Can you show it to me? Did you extract it? It's a zipped folder, right? You need to extract it, I guess. Okay, let it be. Uh, type cd
Yes.
space slash mnt
Yes. MNT. Yes.
slash
Hmm.
slash
One more slash.
press double tab. Tab जो है ना, tab key. Press it tab. हाँ, press tab twice.
Yes, done.
Type C.
C. Yes.
C. हाँ, वहाँ पे you can see C, D and E, right? Type C.
Yes.
Then slash.
Yeah, done. Okay.
Then press tab again.
Done. One more time.
2 times, yeah, as you did in the previous case.
Yes.
Press tab
Okay,
again.
done. Done.
So users, capital U. Tab. It's okay. Slash. After you the slash.
You.
Press tab again 2 times. अब ये कौन सा user है?
Small user. My
Are you sure
system.
it's small user?
Yes.
Okay.
You can
So
see here
small
it's
u.
user.
Okay.
Yes.
Small u
Done.
slash. Then
Hmm.
press tab again. Press tab 2 times. Then desktop.
Uh, Desktop.
Type desktop. Yeah.
Yeah, yeah. Enter.
Press enter. Yes. Type
Okay.
ls. Can you see RNA-seq somewhere? tar.gz.
Yes,
Second
this
last. Yeah.
yes.
Now we need to extract it. ये zipped है. This is zipped folder. We need to extract it. So type
Okay.
tar
PAR.
space
Hmm.
minus
Hmm.
xvzf. I
What,
repeat,
sir?
xvzf
F. Yeah.
space r, small r,
Hmm.
tab, enter. Once you get your dollar prompt back, please let me know.
Okay, sir.
Okay.
So may I share my screen because I'm also not in desktop. I tried the same steps, but I was not still not able to get.
Please share your screen. One second. One second. Okay. Ma'am, can you press up arrow, please? Up arrow key.
Apparently,
Uh,
okay.
Desktop, can you please delete desktop? But wait. Slash users, after users, press slash. Then NAV tab.
And the AV,
NAV, after that, press tab. Now press tab twice. Can you see desktop here or OneDrive?
OneDrive,
There's
there is,
OneDrive. There's OneDrive. Yeah.
yes, yes.
O, capital O, tab. No. Back. Tab. Press tab.
Okay,
Now press tab 2 times.
okay.
Desktop. DESK.
Okay. Again, tap.
Yes. Press enter.
Um, okay.
Type
Okay.
ls.
Okay. I'll
So it's RNA-seq is here, right?
Yes, sir.
Okay.
Do I need to do anything else after this?
No, it's all sorted now.
Okay, thank you, sir.
Now, I hope all of you are on desktop. Let's go step by step. Are all of you on desktop?
Yes, sir.
Yes, sir.
Okay, perfect. Can you see my screen now? I'm taking it as yes.
Yes, sir.
Yes, sir.
Okay, so type PWD. Done? Did you get this path? Did all of you get this path?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. Now type desktop underscore path equals to, and then within quotes, copy this whole thing, paste it here, and then close the quotes. Are we done till here?
Yes.
Are we done till here?
Yes, sir.
Press enter.
Yes, sir.
Done? Are we done?
Yes, sir.
Okay. Now I'm copying a command in the chat. All of you paste it from there. And once you are done pasting, let me know.
Yes, sir. So done.
Done? Okay. I'm copying another command. We are creating a new directory. Copy it from the chat, paste it on your terminal, press enter. Done? Are we done?
Yes,
Yes.
sir.
Does that mean software script folder may we have created a file by the name of RNC?
We have created a folder by the name of RNA-seq. Yes.
Yes, yes.
Yes, yes, yes. Next command is to unzip a folder called MacBook M1 RNA-seq basic environment.tar.gz, and we are unpacking it inside our RNA-seq folder. Okay, press enter.
So this command is for MacBook or for everyone?
It's for everyone. Did you get any errors in between?
No, sir.
No, sir.
It's
No,
happening.
sir.
It's unzipping. Is it done?
Sir,
No, sir.
hello.
Yes, yes, please.
Sir, mine is showing no such file or directory.
Oh,
It's me, sir. Ritupalli.
okay. Can you share your screen, please?
Yeah, sure. Sir. Sir, is that visible? My screen.
Yeah, it is. Um, Project. Are others getting the same error or
Yes, sir. Similar.
similar error?
Yes, sir. Same. It is error is not recoverable. Everything now. It's same.
Okay.
But no
I
such
think
file. Nothing like this coming. Yeah, it's similar.
okay, okay. Just give me a second. I'll send you the command again. I guess there's some issue with the typing. Uh, can you please stop screen sharing?
Yeah, sure.
One second. Okay. There is a small error in the path. Terminal is stuck. Okay, use this command, the one that I'm pasting in the chat. Use this command and let me know if it's working. Is it working now?
No, sir. Error is there.
No, sir.
What error do you see? Can anyone share your screen?
In my case, it's working. I think first command is.
Can you type cd space RNA seq RNA tab, press enter, type ls. Type cd RNA seq tab, enter ls. One second now. Oh, okay. Uh, for the rest of you, did it work? The second command?
Just some commands are keep on running. Is it
Yeah,
that
that's how. Yeah, that's what should happen.
So it's not
okay?
working with.
Sorry.
So it's not working with Mac. Showing error.
Okay, wait. Now,
Okay, sorry.
wait, ma'am. I was looking into their error first.
Okay, fine, sure.
Uh, was it Navneet?
No, sir, mine is the commands
Yes,
are
sir.
running.
Yeah, can you share your
Uh,
screen?
yes,
I'll
sir.
show mine.
Now it's running, right?
Yes, sir.
Okay. Next, whose error? Who was getting this error? Next, who else did get this error?
So me, sir, Prachi.
Share your screen.
So can you see it, sir? Whether I
Not
shared
now,
it.
not yet.
So this one.
Can you type ls? Where is the RNA-seq folder here?
Actually, I entered through a CD into that, sir, earlier. Should I go by that way? CD and then
Mm,
RNS.
Did you do this whole mkdir thing? I can see
Yes,
that.
sir. I did that, sir.
Okay, can you go through CD? ls?
And now LS, yeah. I type tar over here like tar. It's
Can you go one folder back, ma'am?
okay.
Okay. So wait, let me see down. XVF RNA sequence. Okay. Can you press up arrow?
Up arrow. Okay.
Uh, Again.
Up arrow.
Keep on pressing it. Yeah, till you get the tar command. Once again. Once again. Bus. That's it. That's it.
Okay.
Can you just remove one RNA-seq reference based from here? Like
This one.
use left arrow key. No. Left arrow key. Use your left arrow key. After XVF, just delete one RNA-seq. After XVF, delete one RNA-seq.
XVF. Okay, this one. Okay. Acha.
No. ये नहीं. XVF के बाद the first RNA-seq. Can you please delete
This
it?
this one,
Yeah.
no.
Yeah.
Yeah,
Just
that is
yeah.
what I was. Yeah.
Even the slash, delete that too.
Okay.
1 second. Yeah. Press enter from here.
Yes.
Okay.
Thanks.
What about the rest? I think this only error that I can understand is.
Sorry, Mr. Avinash, can I ask you something?
Yes, please.
Thank you. So, sorry, I logged out of the lecture first, few minutes ago. So, have you any analysis to the files that you've sent, or we are just checking the what files are there?
No, we are installing a few tools.
Okay.
So, if you can, just execute this star command that I have shared.
Okay.
And then we will do a small analysis as well.
Okay.
We are
Thank you.
just waiting for this installation to happen. Some of us were facing issues in this. So once we are done, we'll quickly do an analysis.
Hello, sir.
Yes.
Sir, what should I do, sir? I am just.
Are you facing an error?
Actually, sir, I just you told me to list something from that RNA-seq reference based, and I am stuck there. I don't know.
Share your screen, please.
So I will share my screen.
In the meanwhile, can people tell me whether they are done, whether this command like was running for them or not? In the chat, just yes or no.
Sir, can I share my screen?
Yeah, just a second. I'll look into hers and then just 2 minutes. Can you type ls? Can you type ls? Yeah, enter. Okay. Can you type cd dot dot? Type ls again. Okay. Again cd dot dot. Okay. Can you run this command again? The one which I have sent in chat.
MTDAR.
Yeah. Enter. Get into RNA-seq reference based.
Yes, sir.
Okay.
Hello?
1 second, 1 second.
Hello?
Can you listen? Can you hear me?
Yeah, now I can hear
Oh,
you.
can you get into? Okay, if this is done, can you execute this command? The one which I have shared in the chat.
One.
Yeah. Press enter. Okay. Press the up arrow key again. Okay. Uh, just before software script, delete this dollar project folder and type. If you can paste, just paste this RNA-seq reference paste. I have sent it to you in the chat. Just this line, the last one.
Yes.
Yeah. Enter. Now can you run? Can you press?
Okay, then
Wait,
about
wait, 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 wait,
R.
wait. Just a minute. Yeah. Now can you run this command? Press enter.
Yeah, it's running, sir.
Okay. Can you please stop screen sharing?
Yes. Yeah, sure. Thank you, sir.
No worries. Who was the next person? Someone had a doubt in between.
Yes.
Mr.
Yes.
Sabhi, can you please share your screen?
Yes.
Uh,
Can you see? Showing brief.
okay. And did you press enter after this?
Yes, sir.
And what do you get? Just the screen?
Yes, sir.
Nothing changed.
Yes, sir.
Okay. Can you press Control C? Type ls. Okay. I think it has unzipped in by different ways in different folders. So can you get into RNA-seq reference based? ls? Okay. Mm, 1 second. Can you get into CD software scripts? ls?
Yes,
Yeah. Fine. Can you get back to desktop? Hmm. Can you execute this command that I have pasted in chat? I have sent a command in chat.
yes.
Okay. Yeah, it's happening. It will take some time.
Yes, sir. Okay, sir.
Okay. Can you stop screen sharing?
Yes.
Okay, cool.
Sir, even I am
Mm.
stuck.
Okay, who else has an issue?
Sir, me, Shikha.
Yeah, can you please share your screen?
Yes, sir. Sir, nothing happened after this command. I've tried.
Press Control C. Press Control C.
Yes, sir.
Type ls. Then type cd RNA-seq reference. Paste. Press tab because it will automatically fill it.
Mm-hmm.
Press enter.
Enter.
Yes. ls. Okay. See, wait. Get back, type cd dot dot. I have sent a command in the chat. Can you please paste it?
Yes.
No, yeah, there's mkdir,
So,
not all of them. The last one. Yeah. Press enter. It exists? Perfect.
yes, sir.
Perfect. Now, this command, please. The latest one.
Mm.
Press enter.
Yeah, now it's
Okay.
running. Thank you, sir.
Perfect. Anyone else?
Excuse me, sir.
Yes.
Sir, I'm having error. I'm stuck.
Yeah, can you share your screen?
Sir. Is my screen visible, sir?
Not really. I can't see your screen. Yeah, now I can.
I'll share it once more, sir.
Yeah. Can you type ls?
Just give me 1 second. Sir.
Yeah, no worries.
Alice?
Yeah, type ls.
I've done once, sir. Once again, sir.
Okay. Can you go back? Cd dot dot.
Sir, command not found.
Cd space dot dot.
Yes, sir.
Okay. Uh, The second last command in chat. Can you please open it?
This MKD, sir.
Yes.
So file exists.
Perfect. So then can you please copy the last tar command? The last command, tar-xvf.
Yes. Yes, sir, it's working.
Perfect.
Thank you, sir.
Was anyone able to run the whole thing? Did it execute for anyone? You should get your dollar prompt back. Did it finish running for anyone?
Yes, sir.
No, sir, it's still running.
Yes, sir.
No,
Oh, did
sir.
it finish?
Yes, sir.
Okay. In case if it finished for one of you, one second. From desktop, I hope all of you are on desktop. The one who were able to run it. Can you please copy and paste this command? Source. Only the ones who were able to run it. Rest of you can wait.
Mr. Avinash,
Yes.
I executed the command that you gave it to us, but
Okay.
because I logged out of the lecture, so I haven't created the folder called RNA-seq. So all the files have been executed in the folder called software scripts. So can I run this command after I edit it? Like I can delete RNA-seq and directly bin active?
Yes, you can. You can.
Okay.
Okay. Others, those who said yes, for whom the tar command got executed, are you done with the source command?
Excuse me, sir. It's asking to install Xrun. So should I go with it?
Can you share your screen, please?
Yeah, sure. So can you see this, sir? Xrun command requires the command
Yeah,
line
install
developer
it.
to
Install.
install. Okay. Did we type?
What about the rest? A couple of you had said yes. Is the source
Will you
thing,
please
were you able?
share ये बैटरी
My tar
बैटरी
Sorry?
लेने
command.
के बाद ही होगा ये सॉरी थोड़ी देर के बाद देती हूँ चार्जर बैटरी सोर्स चाहिए इसको
Yeah. Anyone else? The last command after source would be conda unpack. Once you're done, please let me know. I'll be back in a minute. Okay. Any any progress? Can you guys hear me?
Yes, sir. I'm facing a problem with the security of the MacBook.
Oh,
I,
I am not too confident about MacBook. I don't know whether if Dr. Jibin is online.
Yes, yes. What's the issue, Mohammed?
so after I use this command,
Uh,
conda unpack,
uh.
saying uh, uh, clear, not opened. So like it's showing a dialog box that there is a malware, and the MacBook couldn't verify if there is no malware in the.
Okay, allowed. You have to go to security setting and give the permission.
Yeah, I did it many times. So I think there are many
Can you share
commands.
the screen one time?
Yes, sir. Is it visible?
Yeah.
Okay. So I'm trying to give this command, conda unpack.
Can you clear the screen?
Yes. It's even with the clear.
Open anyway. Click on open anyway. Use the password.
Yeah, that's exactly what's happening. It's keep showing that there's a malware.
Okay, clear it actually. Clear. First you type clear.
I'm typing clear many times. Where is it? But it's not working. I think I have to. Sorry. I think I have to deactivate the conda environment, the software script.
Shouldn't be. I think. Yes, software script need to deactivate.
Okay.
How it is came actually software script. Open a new terminal maybe.
Okay.
Close it and open it. I think. It's better you keep your file in the desktop actually, otherwise it will create a confusion.
Yeah, it is. That's right.
Go inside software and script. Did you extracted everything?
Yes.
Okay, 1 minute. Okay. Once you installed, 1 minute. Okay. RNA, you are at which one? So inside software script, you say. Type
Yes.
ls rnasic.
So in this, I haven't created this folder.
Why? Should we have followed you? Everything is in a different places actually.
So I logged out.
Bin, etc. Everything is.
But I can like transfer all those files into RNA-seq manually, like this.
It will take time, you see. I think it's better is that inside the software and script, I delete all other things and you start from beginning actually. 1 minute. Go to the terminal. Okay. Delete all other files other than that script and go to the manually, maybe delete it actually.
So I'm gonna delete all those files.
Yeah.
Okay.
Only keep the software, the RNA M1 R1, the compressed files. Yeah, that's keep it. That keep it.
Okay. I have to keep this one.
All the folders you delete it.
Okay. I will.
All the folders.
Okay. Thank you, doctor.
Okay. After that, good.
Yeah, so what's the status?
Mr. Avinash, so I will do it and I will redo the step again and follow exactly what you did. What you mentioned.
Okay.
Thank you.
Thank you.
So it's done, sir. So I'll be typing Conda unpack.
Is it after activating? Can you see RNA-seq in the side, left side?
Yes, sir, I can see.
Okay, then
Can
it's
I
fine.
share?
You done it. You done.
See, like this, RNA-seq should come on the left.
Yes, it's coming, sir. So
Okay. Okay, so I guess so if you can see RNA-seq instead of base, that means it's installed successfully. And
can I share it once so that I'll get confirmation?
yes, please.
Is it fine, sir? So I did Conda unpack and then
Yes, RNA-seq. Yes.
RNA-seq is there.
Yeah. Just type fast QC, FAST QC. Fast QC.
FAST
QC. Yeah.
Okay.
Space.
Space.
Dash help.
Dash.
Help. HELP.
Help. Okay.
Yeah. Enter.
Enter.
Okay. Perfect. Done it.
Okay, sir. So
what about others? Sabir.
Yeah, can others
So
please?
can I also share my screen?
Yes.
Is it fine, sir?
Yes.
No, I think so. This X run or something has to be installed.
Can you type it fast QC? FS the same thing. Fast QC. Space
Then
dash help. Yeah, enter.
Did, sir, but it's showing an error.
Okay, fine.
excuse me, sir.
Yeah, okay. Some issues are there. Okay, we'll solve it.
Avinash sir, Jibin sir,
Yeah.
is it done for today? Because I need to travel an hour to go to home.
Okay.
So it's already 10, it's 9:40. So is it done for today or should we have something?
Yeah, you can join. This thing issue is there. You can join a half an hour before the session. We'll solve it for tomorrow.
No, sir, actually I'm done with this.
Yeah, we are done. We are done with today's
Yeah.
session. In case
We
if
are-
you have any installation issues, you can join and half an hour before yesterday's session.
Yes, yes, sir. Thank you so much, sir. Yeah.
Wait for tomorrow's session, sir.
Okay, sir. Sure. Thank you.
Okay, so those who are getting this syntax error issue, can you, like, this is the last try that we'll do. Sudo apt get install crun. Type your password. Press up arrow. Press up arrow.
Yes,
Remove
sir.
get. Remove get. Just to install. Sudo apt install. Remove get
Removed, sir.
and press enter.
Unable to locate package crun.
Okay. Okay, let's see.
I think we can try tomorrow, whatever it is.
Okay, okay. So
Yes, sir.
maybe tomorrow half an hour before the session. Just before everything, just make sure that before you, like, we'll call it a day today, but
Mhm.
before you shut down your system, just make sure that at least your tar command has run completely
After it
for
has run,
everyone.
I was getting this dollar prompt, so I thought it
Yeah,
is
it's done. It's done. This thing is done.
Okay, sir. I
Yeah,
will
in your
join half an hour before tomorrow's session.
okay. Okay, guys. So just make sure that your tar command is executed, and then tomorrow we can proceed with the installation.
Hello, sir.
Sorry?
Hello.
Yes.
Hello.
Yes.
Sir, after completing the like this is running, so we have to type that source and the other conda unpack. Yeah, we left it like this.
Yeah, conda source and conda thing.
Actually, mine is still running, so that's why I'm asking.
Yeah, you have to run source and then the conda command. Source rnaseq and then conda hyphen
Hello.
unpack.
Hello. Yes, sir. Hello.
Yes, you have to run source and then conda unpack. But in case
Yeah.
you still face errors, then tomorrow, half an hour before the session, you can join and we can fix the errors.
Okay. So
For
we
now,
don't
let
need
the
to shut down
Excuse me, sir.
before this command.
yeah, before the star command finishes, do not shut down.
Okay. Okay.
Yeah.
Okay.
Yes.
Excuse me, sir.
Yes.
Sir, what is this last one which you sent? Sudo apt get install cron.
No, it was for there's a missing package which is stopping this installation. So I thought this would help to install that, but I don't think so. So if you are done with tar, just uh, stop it there and then join half an hour before tomorrow's session so that we can fix the rest of the installation issues.
Okay.
Okay.
Thank you, sir.
Okay. So if your tar command is still running, let it run, let it finish, and then join half an hour before tomorrow's session so that we can fix the other installation issues. Okay.
Okay, sir.
Okay. Okay, guys.
Okay.
Thank you
Thank
so
you.
much.
Thank
Thank
you.
you so much. We'll meet tomorrow. Bye-bye.
Thank you, sir.
Thank you, sir.
Thank you. Thank you. Thank you.